Good morning, students. Welcome to all the students and participants of MOOC course, Introduction to Proteogenomics, Week 7. First, let me thank all of you for your wishes and messages on the occasion of Teachers' Day. It was very enlightening to see a lot of responses and wishes from students from all over India. And I must say that, NPTEL, MOOC course, and different online virtual interactions have opened up new opportunities and new interaction with the students from all over India and different parts of the world, which is quite amazing and very satisfying experience to interact with many bright minds from all over places. So I hope you are able to catch up on the course and able to do assignments. Also, as you know, we are conducting this weekly Q&A session on Skype. Please utilize this opportunity to clarify your doubts and take a slot by filling that detail in the Google Sheet. Also, the link for the reference materials are provided in the video description here. So do take some additional references so that you are clear about these new field and new technologies. The first lecture of this week is going to talk about the need for big data analysis in biology, and especially why it is important to have the data sharing. So Dr. Ratna, he is going to talk about different issues of the big data analysis and how one could overcome that, especially by sharing the correct and unaltered data set, which is very important given that now there is a huge emphasis about data sharing and data upload. It's important also to know that the data formats and how to access those type of data and share with that data set. So role of contributing to big data is definitely important because if you want to really generate reproducible data sets, we need to follow the correct guidelines. And there are some bigger consortium, bigger platforms like Proteome Exchange available with all type of data being deposited from different resources, as you can see on the slides here. And there are some major international initiatives, which is one of them is the Clinical Proteomic Tumor Consortium known as CPTAC from National Cancer Institute and CIA, which is taking a lot of lead now to help community, scientific community, to do the bigger projects with large number of sample and the data being shared by many labs together, being analyzed together, and then come up with the much novel findings. So there are different labs from different parts of US, which is contributing in the CPTAC project. And then they together work on, let's say, different cohorts of the patients, share the data, analyze the data, and then try to come up with what is the novel output from the data set. And the entire data being actually shared to the public community. This is actually coming a little back from the TCG, the Cancer Genome Atlas project, which is started in the last decade, where different cancers' genomes were sequenced, and that data was made publicly available. Idea was, can we take the same samples which was used for the CPTAC, prior to that, the TCGA, and then we do proteomics on that, and therefore over the time period we could also start integrating data from genome and proteome and analyze that data for the proteogenomics analysis. And that's where I think this kind of CPTAC consortium has really played that important role. So you'll be able to get some more detail about the CPTAC unique features which are required in the data portal, different pros and cons of having these kind of unique features, and of course the common data analysis pipelines. Next lecture, Dr. Ratna is going to talk to you about FAIR, which is findable, accessible, interoperable, and reusable. That is the principle for assigning data and also the rule of what is the role of UID for each patient, especially if you're looking at the better and effective treatment modalities. So some of these are very key concepts. And as you understand now, these we are talking about some really bigger projects and bigger initiatives, which is looking for the much more reproducible data set and want to share that with the entire community. So this is where I think you have to start learning these best practices, best skills from the beginning, so that you are better prepared for incorporating those in your actual project, which you are going to start maybe in a few years from now. The last two lectures of the week will be by Dr. David Campbell, who will talk to you about mass spectrometry-based proteomics. He comes from the Institute for Systems Biology in Seattle, and he will first focus on the SWOT technology, which is sequential window for all the theoretical mass spectra analysis, and he will compare that with another targeted proteomics approach, directed reaction monitoring. And then he's going to introduce you to Peptide Atlas and SRM Atlas initiatives from the ISB, which has resulted into some very novel these portals, which is being utilized for targeted proteomic analysis. The last lecture, he is going to talk to you about DIA library QC workflows and also compare the SRM Atlas, SWAT Atlas as well as Peptide Atlas. And while doing so, you will see there is many important criteria for doing reproducible targeted proteomics workflows, looking at the retention time, looking at different parameters, different fragment characteristics. All of those are very important to ensure that the data which you are analyzing and the biomarker which you want to report, those are actually reproducible or not. And again, finally, a lot of statistical models comes into the play to make sure that you are looking at the right candidates, and false negative, false positive could be eliminated out from your analysis. So I hope these are all going to be very interesting and new ideas for you to broaden the your understanding, the overall scope of your learning, and connect you with the much broader initiative which is ongoing currently in the world. And if you do follow the NCI website, National Cancer Institute website, you will see a lot of updates coming from CPTAC. Those are very interesting updates which you should follow it up. And likewise, you should follow the ISB website, looking at some of the new portals which is coming up for different type of Atlas projects. I hope you'll enjoy this week's lectures. For any questions, query, please write in the portal on the query, as well as connect with us on Saturday's Skype meeting. That's all for this week from my side. All the best for doing assignment. Thank you.